It's my pleasure to introduce to you today's speaker. Today's speaker is Amanda Smith. Amanda is one of my graduate students. Amanda has been with us for the last, I would say, almost 2 years, and she's ready to move on with the next step of her life. Amanda graduated from North Georgia College and State University, kind of pretty long name. That's where she got her Bachelor's of Science degree, and then started 2 years ago working with me. We met one time at an ASD meeting where she was very interested in the area of medicinal properties of plants. So when she came, Amanda started working on those two vegetables that she's going to introduce us to. And the title of her topic, or her presentation, is Comparative Analysis of the Antioxidants and Insulin-Patentiating Properties of Bitter Gourd, which is Mijitaba and zucchini. So without further ado, I'm going to leave the floor to Amanda.
Thank you. Dr. Heather, happy faces. My project overall is looking at potential medicinal values of bitter gourd and zucchini. The first plant I looked at is bitter gourd. Its species name is Mijitaba from the family Comparative. It is a tropical and subtropical vine. It grows, bears simple alternate leaves with deeply separated lobes, yellow dioecious flowers that bloom in the northern hemisphere from June to July, and fruit ripen in the fall. This is a picture. Here's a picture of the bitter gourd. It's kind of spiky. Here's a picture of the inside of the fruit with white interior pith and red seeds. They are oblong with a waxy, wart-like exterior. Again, white pith. These plants are consumed in parts of Asia and Africa and Latin America for their value to treat diabetes. They are actually consumed as a food remedy. They can be eaten as soups or stir fries. They have a really bitter flavor that gives them their value. People eat them. Some of the properties that have been researched in bitter gourd are a protein that elicits a hypoglycemic effect in people who eat it. It's called an insulin-patentiating property or an insulin-promoting factor. It also contains triterpenoid glycosides and beta-glucosidase enzymes. The beta-glucosidase enzymes are very important predictor molecules for the presence of some medicinal value. These glucosidases will hydrolyze glycosidic bonds, which is a very important step in other plant processes such as vital hormone regulation or defense against animals that eat it. Bitter gourd also contains charantin and amortacine, which are specific triterpenoid glycosides. They are responsible for giving the bitter gourd its bitter flavor. Another plant that I looked at, I compared back to bitter gourd, was zucchini. Its species name is Cucurbita pepo, also from the same family Cucurbitaceae. Zucchini is actually a variety or a cultivar of sea pepo. Other plants that belong in this species are pumpkins, crookneck or straight neck squash, gourds, plants like that. They are all varieties of sea pepo. Zucchini is a temperate, small bush type plant. It ranges from a very open, wide habit to very crowded, small habit. So its leaves are very wide open, or they're very narrow and tall and upright. They have large leaves, also with deep notches. In the northern hemisphere, it tends to bloom from the May until the frost. They have orange or yellowish flowers. Here's a picture of zucchini. I hope all of y'all know this. Here's a picture of what the cut-up zucchini looks like. They are elongated cylinders. They can vary from a light green to a very, very dark green, almost black. Some of the varieties of zucchini have yellow spots or stripes on them. They are consumed cooked or raw. You can pretty much eat them anyway. They're popular in soups or salads or as side dishes, and hopefully you've all heard of zucchini bread. And while zucchini is not known in North America to be potentially medicinally important, they are consumed in other parts of the world, like in India or Asia, for their medicinal value. Some of the constituents in zucchini are another cucurbitacin, which is another triterpenoid, like what bitter gourd had. Zucchini, on the other hand, has not been shown to have any hypoglycemic activity as of yet. So it's probably not going to have, it hasn't been shown to have any of the insulin-Patentiating properties or the insulin-promoting factors. However, a very closely related plant, Comparative, which is a fig leaf gourd, is currently being used in India to treat type 2 diabetes. So the purpose of my project was to compare zucchini and bitter gourd and just to see overall how well they are at being a potential treatment for diabetes. I looked at their non-enzymatic antioxidant activity. Then I looked at their enzymatic antioxidant activity. I looked at their beta-glucosase activity, which again was that precursor molecule for important plant processes. And then I looked for the presence of this specific protein, the insulin-Patentiating properties. And because in North America they're not really known to be, zucchini is not really known to be a folk remedy for anything, I did expect to see higher properties and higher values in bitter gourd than in zucchini. So the first thing I did, I went to the DeKalb Farmers Market in Atlanta. I bought 3 zucchini and 3 bitter gourd. I took them back to my lab and I put them in a blender and ground them up. I did that to extract the proteins from the fruits. And I extracted each fruit at 0.5 grams of protein per mil of buffer. And the buffer helps to extract the proteins and keep them in their native form so they don't dematerialize or they don't break apart. They're still able to do their things that they're supposed to do. That was one extraction set. Then I went back and got 3 more zucchini and 3 more bitter gourd, and I did the same thing again and had a second extraction set. Here's a picture of grinding the fruits up. I ground them up separately. Here's a picture of the final extract for 1 fruit. So in total, I had 6 bitter gourds and 6 zucchini. Just like that. For my results, I used a randomized complete block ANOVA to determine to compare the two at 95% confidence levels, and I used Turbostat to run the results. After I extracted the proteins, I had to find out how much proteins I actually was able to extract from the fruit. So I did Bradford method, which quantifies the amounts of proteins in my extracts. And after I found out how much they were, within each extraction set, I diluted them all down to a constant level. So extraction 1, I diluted everything down to 1.0 milligrams of protein per mil buffer. And then extraction 2, I diluted them down to 0.4 milligrams of protein per mil buffer. And they're different levels because they're different extraction days. Things happen. You're not always going to get the same amounts. So I went to the smallest amount of protein that I had in all my samples for that extraction set, and I made them all constant. I ran a gel electrophoresis, and these are some of the proteins that I got in the bitter gourd and zucchini. Bitter gourd's on the right, zucchini's on the left. This is my standard showing the size of the proteins that I was able to get. And graph showing my combined extracts. And both extracts combined show that there was a big difference between bitter gourd and zucchini and the amount of proteins that I extracted. However, when I ran the results, when I did my ANOVA, I found that there was no significant difference between the amount of proteins that I extracted. So even though my graph shows that there's a pretty big difference, in reality, there's not. Just how it goes. Total phenolic content is important because it's a very wide class of compounds in the plants. You have tannins, chlorophyll, what else? There's a lot of products in plants are usually phenolic compounds. Gallic acid is also another one. So I wanted to see how much bitter gourd and zucchini had of those compounds. And I used a full-in CO calcium method using gallic acid, which is a phenolic compound, to compare back and see how much my plants had. And I couldn't determine which compounds they had. I just assumed that it was a phenolic compound and compared it back to gallic acid, and just assumed that what I had was equivalent to so-and-so micrograms per mil of gallic acid. So I put my plant extract in with this reagent, full-in CO calcium reagent. After 5 minutes, I added some sodium bicarbonate to it just to complete the reaction. And then after an hour and a half, then I took the absorbance of it. So I put it in a spectrophotometer, and I measured the amount of light that was absorbed by the extracts, and that gave me a value that I can compare back to determine how many total phenolic compounds were in this extract. Overall, bitter gourd had 5.57 micrograms per mil, equivalent to gallic acid for extraction 1. Zucchini had 3.10 equivalents of gallic acid, 3.10 micrograms equivalents of gallic acid. Extraction 2 showed the same trend, 7.70 of bitter gourd and 5.50 of zucchini. So these are the combined extracts again. Bitter gourd did have more phenolic compounds than zucchini. When I ran my ANOVA, I found that there was a significant difference. So bitter gourd does have a significantly higher amount of total phenolic compounds than zucchini. My F ratio was 1.92 compared to 161.4. The third thing that I looked at, which were non-enzymatic antioxidant activities, was the ability of the extracts to scavenge free radicals. Free radicals in your cells or in your tissues can cause damage. They can cause premature aging or wrinkles or cancer. So you don't really want them in excess in your body or even in the plants themselves. The free radical that I used in this assay was 1,1-diphenyl-2-picrolyl-hydrazine, or DPPH for short. And I compared the ability of my extracts to scavenge these free radicals was enzymatic antioxidant activity. Enzymatic is different from non-enzymatic because non-enzymatic is just what properties you have in the plant that are not proteins, they're just like molecules. They'll scavenge things through their electrons. Enzymatic antioxidants will bind to something in your body and neutralize it that way. And the enzyme that I looked at was superoxide dismutase. I used the superoxide dismutase, or the SOD assay, to measure the amount, or measure the reduction of some free radical, or this NBT. I put my plant extracts in this buffer, and the buffer created an environment that would cause a buildup of O2 negative. Excess O2 negative in your cells can cause oxidative damage or oxidative stress. Also causes premature aging, wrinkles, cancer. So you don't want these in your body, you don't want these in the plants as well. So the plants, because they also can build this up in their tissues, plants have to have some enzyme to get rid of it all. Otherwise they'll cause damage to them. And it will cause benefits for people to eat it. So I put this extract in here, in this buffer, and after 10 minutes or so in a growth chamber, put it in highlight area. I measured how much the amount of O2 negative was decreased. I drew the magically. Found that opposite to what everything else has been showing, that bitter gourd has more activities. In this case, zucchini had more ability to get rid of potential oxidative damage than bitter gourd. Zucchini had 1.38 units of activity of this enzyme per mil. Bitter gourd had 0.99 units of activity. That trend was the same to a lesser extent in extraction 2. Zucchini was higher, but not to the same extent. Here's a graph of the combined extracts. Zucchini, in this case, does have more scavenging activity of this, enzymatic scavenging activity of this. Okay. However, in the results, there was no significant difference between bitter gourd and zucchini. So, at all, even though zucchini had more overall activity, it was just the same amount as bitter gourd. Another thing that I looked at was beta-glucosidase activity. That beta-glucosidase is an enzyme that will hydrolyze glycosidic bonds, and that again is an important precursor to other processes later on in the plant, such as phytohormone regulation or scent production or defense against herbivory. So these again will predict some potential medicinal value. They're fairly common across kingdoms. Eukaryotes have them, plants have them. Pretty much any living thing will have beta-glucosidase enzymes. So I put the extract in a test tube with the preferred substrate of my enzyme, and assuming that my extract had these enzymes in them. And after an hour, I had another reagent to stop the reaction, and then I measured them spectrophotometrically again to determine how much beta-glucosidase enzymes are in my extracts. I found that bitter gourd had 0.21 units of activity of this enzyme compared to zucchini, which had 0.09. Extraction 2 was not as evident, you couldn't see the trend as much. 0.11 units of activity of this enzyme compared to 0.1 for zucchini. The combined extracts show seemingly a big difference. This is expressed in the units of activity of this enzyme. And I did not find a significant difference between bitter gourd or zucchini, however. So, they do have these enzymes in them. Just, if you were to eat them, there's not really going to be any difference. You can eat either one, you get just the same amount. No protection. And the final thing that I did was to look at the presence, or determine the presence of my insulin-potentiating properties in my extracts. Again, these are responsible for, or supposed to be responsible for eliciting a hypoglycemic effect in people who eat them, because bitter gourd is consumed as a folk remedy for diabetes. This is what I was thinking was in the plants that gave them this ability. So I used a Western blot analysis to determine the presence of that protein, using antibodies against this specific protein. I used nitrocellulose transfer membranes, non-fat milk as a blocking agent, and NBT to look at the color development and see if the protein was there. I did this twice. It didn't really work out that well. The first time I tried it, I was able to transfer my proteins onto a gel, and then transfer the proteins in the gel onto the transfer membrane. And then when I added the antibodies and did the whole process, I was not able to see the presence of this protein. The second time I did this, same thing. Proteins were transferred onto the membrane. I just did not see this specific protein in my extracts at this time. So I'm not able to deny, but I'm not also able to, neither am I able to confirm the presence of this protein in my extracts, which goes against previous studies. A lot of studies have shown that bitter gourd do have these proteins in it. So I'm thinking that I did something wrong in the process, that maybe I used the wrong antibody or just did something trivial that had a very big result. So we're going to go back and try this again and go through step by step to make sure that I didn't do something wrong. And hopefully I can find it. Here's a picture. It's kind of hard to see, but here's a picture of the transfer membranes. If there were proteins on here, you would see bands running horizontally somewhere in here across the picture. This is the one for an antibody against insulin, and here's an antibody against my specific protein. The insulin I did just to see, just for the heck of it. But there was no bands present. I'm thinking, I think I did something wrong there. But in conclusion,
between 20.
Between 20.
Clean, okay, that's pretty mild wash. All right, so it shouldn't have stripped the proteins off. It might just be the antibodies simply will not react against plant IPP.
Any questions? Yes.
When you were looking at your extracts, you were using your raw extracts each time you were putting those in there, or were you doing any purification?
I centrifuged them down, got rid of all the junk. and just-
But that was pretty much it. So I guess what I was thinking was when you were looking at your chlorophyll in particular, how much gallic acid, your chlorophyll content, that grouping there, I was thinking, was there anything in there that might interfere with the spectrometry?
It's a good possibility. I think phenolic compounds have been, they're very hard to track down because they do have, one of the properties of them is that they will confound results. But that's what I've read. Trusting that it didn't.
Any other question? Well, thank you very much, Amanda. Again.